Hello dear students, welcome back to Karim's Biology. So we have launched a new series called Competitive Biology, in which we will be giving 3 questions in each session, which we will discuss elaborately and we will try to give the answer. Okay, now let's come to the first question. The first question is about the chloroplast. It is about the DNA present in the chloroplast. So the DNA present in the chloroplast is, this is the question that was asked in NEET 2024. Now let us look at the options. The DNA present in the chloroplast is linear double-stranded, circular double-stranded, linear single-stranded, circular single-stranded. Now you may be wondering why the DNA present in the chloroplast. There are 2 cell organelles present in the eukaryotic cell which have a DNA. What are they? Chloroplast and mitochondria. Then why the DNA present in these 2 cell organelles? From where this DNA came? So to answer these questions, there are a number of theories proposed, but one of the most important and famous theory that was proposed to explain this is the endosymbiotic theory. The endosymbiotic theory states that millions of years back, there was heterotrophic eukaryotic cell, and this cyanobacterium, which is having the chlorophyll, it has entered into this heterotrophic eukaryotic cell, and after entry into the cytoplasm, it started helping the heterotrophic cell in providing the nourishment, and in return the heterotrophic cell was providing it shelter. Okay, so this type of symbiotic association has been carried out for several millions of years, and after some time the cyanobacteria that entered into this eukaryotic cell, it has now become the integral part of the eukaryotic cell, and later on it continued to survive within this eukaryotic cell. The same theory can also be applicable to mitochondria. That is the reason why, as they are cyanobacteria, they have their individual DNA. They have their DNA in the form of a plasmid. They have circular DNA which is double-stranded in nature, right? So this is the diagram which shows that DNA, which is double-stranded DNA. There is nuclear DNA present in the cell. In addition to the nuclear DNA, mitochondria and chloroplast, both of them, they have their own DNA. So the presence of their own DNA makes them semi-autonomous in nature. That means independent of the cell, they can divide by themselves, they can increase their number, right? So that is the reason why they are said to be prokaryotic cell in eukaryotic cell, because the nature of this chloroplast is somehow similar to a prokaryotic cell. For instance, the chloroplast has DNA in it, which is circular in nature, and the chloroplast also have ribosomes which are of 70S type. So these are the 2 characters which makes the chloroplast similar to a prokaryotic cell, and as they are living in the eukaryotic cell, eukaryotic plant cell, they are said to be prokaryotic cell within eukaryotic cell. So the answer to this question is circular double-stranded DNA, right? Now let us move on to the next question, which is very interesting. So this is the question about the replication of DNA in E. coli. Which of the following statement is correct regarding the process of DNA replication in E. coli? So this is also another question that appeared in NEET 2024. Now let us look at the options. The first option is the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 3' to 5' direction. So the first option is stating that DNA dependent DNA polymerase, it only catalyzes the polymerization reactions, that is the polymerization of the polynucleotide chain only from 3' to 5' direction. And the second statement states that the DNA dependent RNA polymerase catalyzes polymerization in one direction, that is from 5' to 3' direction. Now the option C, the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 5' to 3' as well as 3' to 5'. So the third statement is actually stating that the DNA polymerase can catalyze the polymerization of nucleotides from 5' to 3' as well as 3' to 5'. So the fourth statement, the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 5' to 3' direction. So what could be the answer? Let us now have a discussion about this. So this is the DNA strand, So this is the DNA strand. You can see this is the strand which is acted as a template strand, and there is one more strand that is being synthesized newly by using this template strand. Okay, so here you can see that there is a template strand, and the opposite strand, which is also called the complementary strand, is being synthesized here with the help of the DNA polymerase. And you can see this is the 5' end where the phosphate group is attached, and to the other side, that is towards the newly formed nucleotide, the newly attached nucleotide, here there is 3' end. Okay, so the polymerization always happens from 5' direction to 3' direction. Why? Because at the 3' end, that is at the third carbon of the ribose sugar in a nucleotide, there will be a presence of free hydroxyl group. The free hydroxyl group is highly reactive, whereas the 5' end, it has already a phosphate group attached to it, it is not free. Only the 3' end is free. That is the reason why the polymerization always takes place from 5' direction to 3' direction. Okay, so I think you are aware that in the process of DNA replication, there are two types of strands that are formed, newly synthesized strands. One of the strand is being synthesized continuously, that is, it is polymerized continuously from the 5' end to 3' end. There is no interference. Whereas in the second strand, there is no continuation of the polymerization. Okay, it happens in the form of segments. Later on, all the segments will be attached to each other with the help of the enzyme ligase. That makes it two strands. So the strand which continuously synthesizes, that is called leading strand, and the other strand which is discontinuous, which is synthesized in the form of small segments of DNA, this is said to be lagging strand. And these small segments of DNA that are formed from 5' to 3' direction, they are said to be Okazaki fragments, which later on will get attached to each other with the help of the ligase enzyme. So the correct answer is the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is from 5' to 3' direction. Whereas you can see that option B, it was given that the DNA dependent RNA polymerase catalyzes polymerization in one direction, that is from 5' to 3'. Okay, so you may think that as it is given as 5' to 3', this is also a correct answer. But actually in the statement it is given that the DNA dependent RNA polymerase, this statement is about the RNA polymerase. It is about the transcription process, but not about the replication process. So the correct option is always D, right? Now the third question. Following are the stages of cell division. He has already given the stages of cell division, but they are in a zigzag manner. A, gap 2 phase, B, cytokinesis, C, synthetic phase, D, karyokinesis, E, gap 1 phase. Choose the correct sequence of stages from the option given below. Okay, so these are the stages of the cell cycle. So you have to arrange these stages in a proper order. Okay, so you know that in the cell division process, there are cycle of events that occur in the cell division process. We call that as a cell cycle. So here the cell cycle is particularly divided into 4 stages. First one is G1 phase, second one is S phase, third one is G2 phase, and the last one is mitotic phase. If it is meiotic division, the last one will be meiotic phase, right? So what happens in these stages? In first phase, in G1 phase, normal cell growth and development happens, okay, and the number of and in the G1 phase itself, the enzymes required for the replication of DNA will be synthesized. Whereas the second phase, which is the S phase, the name itself indicates that S phase stands for synthetic phase. In synthetic phase, the DNA replication happens. It is one of the most important step in the cell cycle, because without the duplication or replication of the DNA, the cell cannot go for cell division. So the important step of DNA replication happens in S phase. And the third phase is G2 phase, and last phase will be cell division phase, which is mitotic phase. Within mitotic phase or meiotic phase, again there will be 2 stages. In the first stage, the nucleus will divide. First of all, the contents of the nucleus will divide. Later on, the contents of the cytoplasm will divide. So the division of the nuclear contents is called karyokinesis, because karyo stands for nucleus, and the division of the cellular contents, it is said to be cytokinesis. So the correct option will be here. First stage will be gap 1 phase, which is G1 phase. and the second step will be synthetic phase, and the third phase will be gap 2 phase, and the next phase will be the division of nucleus, which is karyokinesis, and the last stage will be cytokinesis, which is the division of cytoplasm, thereby completing the process of cell division. So here the correct order here will be E, C, A, D, B. E, C, A, D, B. So the correct answer will be option 4, E, C, A, D, B. Thanks for watching. I will meet you in my next session.